Hello and welcome back. This is part 3 of the lecture on chapter 16, adaptive immunity. We had just finished talking about antigen presentation. We're moving now into, we'll talk about T cells first, and then we'll talk about B cells, and then we'll be a wrap. So T cells, the T lymphocytes, these are cells that, like all the blood cells, sorry, all the white blood cells, they start out in the red bone marrow. But then what's special about these is they travel to the thymus gland where they get a little more education on fighting things, fighting infection. They circulate in the lymph and in the blood. Remember, the lymph is the fluid, just like plasma, that's leaked out of the blood vessels, but it's being recollected to bring it back to the heart. So they migrate, they move intentionally to the lymph nodes. They hang out in the spleen, the Peyer's patches, those areas in the digestive system. And they have receptors on their cell membrane. Remember, these are like the cops that are looking at the license and registration, but they can only see the license and registration if it's presented in a certain way that their receptors can bind to. And that's the so-called T cell receptors, TCRs. So if you see CR, it's cell receptor. So the specificity of these T cell receptors, they don't recognize those little bits of the antigen directly. They have to be presented on that major histocompatibility protein. It has to be presented on that platform, or else this cop cannot see the license and registration. The T cells are most effective against pathogens that are already inside a cell. The T cells don't do great with stuff that's in circulation. And again, why? Because it has to be presented to them on a platform. And if it's in circulation, nobody's presenting it on a platform. And the T cells can also act against body cells that maybe aren't infected, but are still producing things that don't look quite right when they're on the surface. All right, so the T cell receptor, TCR, again, it's a structure that's embedded in the cell membrane and it sticks out to the outside. And then there's this particular place where it can bind something, but it can only bind things that are being presented to it. We talked about the cytotoxic T cells. These are the ones that can actually, this is the cop that goes right up to the cell and checks. And it can, if it sees a problem, it can actually directly kill the cell. And then there are the so-called helper T cells. Those are really the boss T cells that they help to regulate what the cops are doing. And then so the regulatory T cells or T lymphocytes, these repress the adaptive immune response. So once an infection has been cleared, these are the cells that kind of tell everybody, settle down, stop reacting to things. We don't have to make more copies of ourselves. We're done. And actually, okay, so this is an important point. This is a deletion. So when the immune system is forming, when a fetus is in utero, you start having the cells first forming. Some of these cells are duds. Like some of these T cells that are being made, they are supposed to go up to those major histocompatibility proteins, and they should be able to bind to it, right? If they can't find the platforms, if they can't find the very thing where the cells are all going to be presenting their stuff, then they're worthless. And so if a T cell cannot bind to this major histocompatibility protein, then it just killed off. It's like, no, we do not need copies of you because you're no good to us. Then there's a second test, excuse me. Then how can I put this? It's like the cell is tested, and if the cell is too blind, like it can't even see the major histocompatibility protein, then it's not going to be helpful. And so it gets taken out. But it's got to be a little bit blind because it's saying, okay, we need you to see the major histocompatibility protein, but we need you to not react to most of the stuff that's on the major histocompatibility protein. So don't react to like normal driver's license and registration. Don't attack that. And so if it starts reacting to normal driver's license and registration, then it's killed off. So we need it to be not completely blind, but a little bit blind to things, to not go after our own tissues. If you allow this thing to survive and go after its own tissues, that's how you get an autoimmune disorder. So yeah, so there's some editing, shall we say, that happens in the early stages of life to make sure that the baby's immune system isn't going to be attacking its own tissues. In those first few weeks of being out of the uterus, there's a whole lot of learning that goes on, and the immune system is still learning what is self and what is not self. Okay, now let's move on to the B cells. These B lymphocytes, they are, again, made in the bone marrow and mature in the bone marrow. They're found primarily in the lymphatic structures like the lymph nodes, the spleen itself, the malta, mucosal associated lymphatic tissue, like in the digestive system. Some of them can be found in circulation. And when they are matured and able to secrete antibodies, when they've been activated to make antibodies, that's when you'll hear them called plasma cells sometimes. The B cells, like the T cells, are also pretty specific in what they can react to, determined by their cell receptor. So a B cell receptor is a BCR. Each B cell has a bunch of copies of the BCR. So a particular cell is going to react to a particular structure that's shaped a certain way. And all of the BCRs on the same B cell, all of the B cell receptors on that one B cell are going to react to that same one thing. But the B cell over there has a bunch of B cell receptors, but it's going to be sensitive to a different structure. And that's how you get specificity. And the same went for the T cells, and I didn't make enough of a big deal about that. So each B cell generates a single kind of B cell receptor. It's going to go after a particular structure. Let's see. Yeah, so there's these antigen binding sites. Let me show you on a diagram. And each B cell receptor recognizes only one particular shape. Now, an individual human being, so the entire repertoire of an individual's, all the different B cell receptors of the different B cells that we have in our body, they're capable of recognizing millions of different tiny little structures. And it's when we get infected with a particular thing, a B cell will bind to it. And once it binds to something that's not supposed to be in our body, that B cell starts making lots and lots and lots of copies of itself. We call that clonal expansion. And I have videos about this in the homework. So then here is the B cell receptor. It's embedded in the cell membrane like before. And then this little tip over here, this is the thing that's really specific. We call it the variable regions because different B cells are sensitive to different things. But within a particular B cell, all of these structures are going to be shaped exactly the same way. Okay. So then, so these little shaped things, that is the same structure that you're going to see at the end of the antibodies. That the arrows that the B cell is going to be releasing, they're going after anything that's shaped just like this thing. And it's going to make thousands and thousands of these very, very quickly. And a cell that's going to make a lot of these proteins called antibodies and going to ship them out. Remember what that entails at the biological level. It involves transcription, doing translation. So you're going to have a bunch of ribosomes, and those ribosomes are going to be studded on the outside of the rough endoplasmic reticulum, and you're going to have a whole lot of rough endoplasmic reticulum. So let's have a look at what a plasma cell looks like. Here's the nucleus on the inside, and then do you see all these like layers and layers and layers? All of that is rough ER with ribosomes studded all over it. So this thing is cranking out those proteins. And so it's needing a tremendous supply of amino acids so it can put them together in the right order, excuse me. So it can shoot out all of these little protein arrows called antibodies. Just so beautiful. So transcription happening like crazy here inside the nucleus, and then translation happening on all those little, I wish I could make that bigger. All those tiny little ribosomes are actively putting together the amino acids in the right order so it can shoot them out using the Golgi apparatus, the Golgi bodies, to ship those out of the cell and release all of these thousands and thousands of antibodies within seconds of each other. Okay. Basic antibody structure is like the B cell receptor, kind of Y-shaped, and it's here at the end that it's very specific, that it's going to bind just a particular shape. So this is the really stylized diagram, and then this is a slightly more realistic version that's showing the alpha helices and the beta pleated sheets, and then the three-dimensional folding on top of that. Functions of antibodies, so that the very tip of them, that's the bit that's going to be real specific to what it's going to bind. So a few things that it can do, it can neutralize, that is, it can just attach itself to a toxin or to a bacterium, and it makes it harder for that structure to do damage to our cells. It does not, those antibodies are not destructive. Like it doesn't immediately kill this bacterium, and it doesn't immediately destroy this toxin. What it does is neutralize it. And I'm going to give you an analogy, a way to think about this. If you've ever seen newborn babies in the early days, they have like zero control over their hands, and they're forever scratching themselves because their little fingernails are so sharp, and so the parents, in an effort to keep the kid from like harming themselves, they don't like cut the fingers off. They put a little bag, like a little sock, hand sock, little mitten around the kid's hand, so he's still like flailing, but he's at least not scratching the mess out of his face. That is how I picture these antibodies, just binding, making it so that this thing cannot do damage. It's not going to destroy it immediately, but at least it's going to neutralize it. Then these things, remember we said they opsonize, they make tasty, make tasty for the phagocytes. So it's easier for the phagocytes to grab a hold and eat. And there's agglutination. These things are not like a single. I'll show you another antibody that actually has more connections on it than just two. It actually has 10. And so it's easier to like bind more of these things together, which makes it easier for the phagocytes to capture more of them and get them out of circulation. All right, some classes of antibodies. We're not going to go into a huge amount of detail. However, there is a video in the homework, two videos. One has to do with primary response, and the other one has to do with secondary response, and it addresses the fact that there are these different classes of antibodies. The two that I'm going to focus your attention on the most are IgM and IgG. Let me point something out. So a couple of things. Antibodies are also known as immunoglobulins. immune system, globulins, a globular protein. So Ig is where immunoglobulin, where the Ig comes from, is immunoglobulin. And then these letters, M, G, A, and E, the easiest way, the easiest mnemonic to use for this is GAME, G-A-M-E. Those are the four classes of antibodies that I need you to know. And the two I want you to know the most are the two consonants, M and G. IgM is massive. It's a really big one, and it's the very first antibody that's produced when one is exposed to a microbe. It takes about 5 days to get any detectable amount of IgM in the bloodstream, and it takes up to 10 days to see any detectable amount of IgG. The first time, the primary exposure. So that's a very slow response. It takes a while for the adaptive immune system to respond to something. But then the secondary response is insane. The IgG goes through the roof faster than we saw IgM going up the first time. So at about day 4 or 5, we start seeing this massive increase in IgG. And so if it's helpful to you, think gigabytes of memory. This is the one that's more associated with memory. You get this big flush of it. Okay, and I'm going to show you a diagram that shows you a little bit of shapes, but I'm not going to go in a huge amount of detail. Longest lasting antibody. And I will tell you about this one, IgA, associated with body secretions. Think aqueous solution, like breast milk. You've ever heard that breastfeeding is good for the baby's immune system? It's not mysterious. It's this. There's IgA in breast milk, and when baby drinks breast milk, some of those antibodies rush up into the ear canals and easy to fight infection. So it's the kids who are breastfed get ear infections less often than kids who are not breastfed. No mystery here. Okay, last slide is these general shapes. I just wanted to point out IgM is huge. It's massive. M for massive or mega. It's the one that's got like 5. It's a pentamer. It's got 5 of these antibodies kind of stuck together, which makes agglutination really easy. And then IgG, this is the one that we see in huge, huge, huge numbers on secondary infections. In fact, the second time somebody is exposed to a microbe that they've seen before, sometimes they don't even have symptoms. They fight off the infection so effectively that they wouldn't even have known that they were exposed. And then finally, IgA. Oh, let's see. Go back to a second. That secondary response, that is the thing that we're trying to induce with vaccination. You do the initial exposure, hopefully the person doesn't get really sick. They might have a little bit of symptoms, headache, maybe a little fever, a little bit body ache, but they're like, they don't have to be hospitalized or anything. And then if they're exposed for real to the microbe, you have so many IgGs produced so quickly that the hope is that the person just never develops the illness. Okay. And then that last thing that I was going to tell you is IgA. Sometimes looks like this. This is the one that we see in fluids of the body, the aqueous solutions like breast milk, tears, saliva, mucus. So in the respiratory system, for example, it's in the mucus, helping us to fight off respiratory infections. All right, and that is the end of the material on chapter 16. Hope you have a great week, and I'll be in touch about the next exam.